So next up is Dane Wittrup. Dane is the CP Dubs Professor of Chemical and Biological Engineering at MIT and member of the Koch Institute. Dane invented this incredible method called yeast surface display that allows you to really engineer proteins that have desirable properties. His lab has been really using this platform along with his expertise in protein engineering to develop very interesting immunotherapies. And so I'm really looking forward to hearing about your work, Dane.
Thanks, Lori. So I'm going to tell you about a project that we're working on. It's basically a protein engineer's version of bioconjugation. You know, we're all talking about what our backgrounds were and why we approach things in certain ways. So when I think about associating a small molecule with an antibody, I think I'll use an antibody binding site to do that. So if you have a bispecific antibody with one, component of it, one moiety targeting surface receptors in the cells you'd like to reach, and another one just capturing and holding very tightly to the drug that you want to deliver, that's what we're looking at delivering. Now, this would not be a good idea for some of these very potent cytotoxics we've been discussing so far today, because any small amount of leakage could be problematic. But there are large classes of molecules that this is just right for. So for example, if you have something that has poor PK but is well tolerated, like, say, the PROTAC drug class, it might be nice to be able to drive by this receptor-mediated endocytosis uptake into particular cell types. And so the potential advantages in the example of PROTACs is they have common handles on them to bind to the E3s. You can actually block off some of the hydrophobic surfaces of these drugs that give them such poor PK. This could actually be developed for any drug that you can make an antibody against. And again, from the protein engineering point of view, we don't have to do any chemistry. So we just make another protein. So I love listening to you guys talk about what you do, but I can't do it. So. Also unproven yet, but kind of intriguing, is the idea that one can get a higher Cmax intracellularly by release, pH-mediated release from an antibody than you might from, depending on proteolytic release. And you can also play around with, and we've done some of this, cocktails of mixing and matching on a given capture antibody. So I mentioned that we were doing PROTACs first because essentially the great majority of commercially developed PROTACs use one of these two classes of E3 ligands, either a thalidomide derivative or VHL1. And so using yeast display, which is our favorite tool for such things, we wanted to make very tight binders to these molecules. Here is some flow cytometry yeast display data. On the y-axis of these dot plots is a signal for binding to the target, the top row being VHL and the bottom row being thalidomide. And after a long time of competition, in this case about a day, you see that it hasn't dropped that much. With the thalidomide, after 6 hours, it's dropped more, so we still have some work to do to make that a tighter binder. But I'll show you some more of the data on the VHL1 now. Here, once we reformat it off of yeast into a soluble IgG, we find that at 37 degrees, the dissociation half-life for binding to the VHL1 E3 ligand is about a day. So if we load this onto a bispecific containing these binding sites, we'll hold on to it for many hours to days. So first looking at loading up these antibodies with different actual PROTACs, you find that at the concentrations where we're preparing these stock PROTACs, within about an hour, we've fully loaded the bispecific with the PROTAC, and these are two in particular that we're cocktailing together and using. One of the questions is, what will release look like intracellularly? So if you shift the pH to lysosomal pH, you lose about 1,000-fold on the affinity, so it's reasonable to expect we'll get prompt release once taken up and fully trafficked, and with some potential release along the early to late endosomes as well. Here's an uptake experiment in culture where we did two different fluorophores, one for the antibody and one for the probe, the VHL, and then it was just linked to a fluorophore. And when we just pulsed it on, you could see not much got taken up. But if we incubated in the presence of the antibody, we had progressive uptake of the antibody, and this was an integrin-targeted antibody. And here on the two-color flow cytometry, we show the correlation, a pretty good correlation between how much antibody was taken up and how much probe was taken up, consistent with the great majority of the uptake being driven by that endocytic route. And then just as a control, in the absence of any antibody, a certain amount was taken up as well, but this was after sitting for 2 days soaking in the high concentration of the drug, which would not be something available to you pharmacologically. We did a little bit of modeling to see whether or not we could hit the necessary affinities to actually get efficient delivery. And first off, what sort of exposure, systemic exposure, could we get? So we just simulated trajectories accounting for if the drug were to dissociate, it might rebind back onto the bispecific, but it might also be captured by serum proteins. And so we looked at various different regions of that parameter space and came up with this map of it, looking at dissociation rate constant on this axis and association rate constant on this axis. And the PROTAC alone had a certain area under curve for the systemic exposure. And the two binders that I already showed you data for put us up here in about a 10-fold or more increase in systemic exposure. So that's interesting and worth pursuing. So here is a two-color experiment in mice where we've preloaded on the antibody. It wasn't a bispecific in this case. There were no tumors. We were just to see if a circulating anti-payload antibody would retain it for a long time. And so looking both at the antibody signal and the loaded probe signal, you see that out to a couple days or so, you still have appreciable amounts of payload being carried. Whereas here, of course, the clearance of the payload on its own in the absence of the antibody is really just minutes to an hour or so. So it's consistent with this idea of getting at the very least an order of magnitude improvement. So we're gearing up now to do a therapy trial with this. In the mouse systems, the mouse tumor systems, the syngeneics, there are not, in general, a lot of useful anti-tumor antigen antibodies. The TA99 antibody in the B16 melanoma is an exception. But a few years ago, we did find, interestingly, that essentially all of the widely used syngeneic murine models have as a very immunodominant surface antigen an ONV protein from an endogenous retrovirus that's epigenetically altered and upregulated to be expressed in these tumors. And so in that publication, we also showed through the cryofluorescence tomography experiment that we would get pretty strong localization of an antibody against that ONV protein to the tumor, and this is just probe from the antibody that's been released. Interestingly enough, these targets are also present in humans, and there are at least a couple of startups pursuing immunotherapies towards these ERV antigens in human cancer. Okay, the high-speed delivery has brought us back onto time. And so our next step, that wasn't an accident. I don't always talk that fast. So we're going to bring together these payloads and the targeting and see what we can do on a syngeneic tumor model. And we like doing the syngeneics because we expect there will be an important immunotherapeutic component to any actual response. And all the work I've shown you was the work of a chemical engineering grad student, David Crum, in the lab, and we're collaborating with Eric Fisher from Harvard Med. Thank you.
Questions?
First question will be, what? No.
No. We got him. You just got to give.
Yeah.
Would there be any advantage in using the technology for cell impermeable drugs?
Would there be an advantage for using the technology for cell impermeable drugs? Impermeable. Yeah, I guess the idea being once dropped off, they would persist only in the cell in which they were internalized, as opposed to if we're getting the endocytic delivery and they diffuse right out, there's less of an advantage.
Thank you very much for a great talk. Does your binder works for all PROTAC with the VHL warhead?
Yeah, so all the PROTACs that had VHL1 also bound to the antibody, and all the other antibody, all the ones that had thalidomide or a pomalidomide also bound.
And so there are different VHL warhead that with a similar but slightly different structures. Does the same binder works for all of them?
Yeah, if David was here, he could tell you. But I don't think we have that problem so much as we do with some of the thalidomide variants. With the VHL, I think some of the more, current generation VHL1s, it still does work on.
Thanks.
And if they didn't, you could always engineer them to do so. But
Okay, one last question.
That was my question, so I'll ask another one. What do you think the optimal non-covalent affinity range is at pH 7? Like, is it single-digit nano, or does it have to be higher or lower?
I mean, I think of it really more kinetically than in terms of actually equilibrium, because we preload it and then put it in, and then it'll dissociate, and there may be some recapture. But I just go in terms of time scales of that dissociation, and compare that to how long it takes to accumulate in the tumor, and how long it'll take for the antibody to clear. The antibody will be there for a long time, and so some balance will be struck there with respect to how much toxicity would you get from leakage off in circulation. By which I'd say, I don't know, but we're trying a day right now.
Great, thank you.
Yep.
Okay, let's thank Dane again.